Something happened in your body that no one fully explained to you, or if they tried, the explanation they offered was a clinical description of a biological countdown, the ovarian reserve depleting, the hormonal signals quieting, the reproductive architecture stepping back from its decades of active service. That explanation is technically accurate. It is also spectacularly incomplete, because what it describes is the mechanism. What it entirely omits is the purpose, and the purpose, the specific evolutionary reason that this transition was built into the human female body with genomic precision and maintained under active selection pressure for at least 300,000 years, is the most consequential piece of biological information about women that modern medicine has consistently failed to transmit. This script is the transmission. Set aside whatever the day placed on you, the particular weight of being responsible for things and people who depend on you, the professional precision you sustain for hours without acknowledgement, the continuous low-level calculation that constitutes life as a woman of experience in a world that does not yet have adequate vocabulary for what experience in a female body actually produces. Set it down. Not because it is not real, because for the next several hours something more foundational is worth your attention. Something that will change the frame through which all of it looks. Begin with a number: 51. The average age at which menopause occurs in modern human populations, confirmed across population studies spanning continents, cultures, and millennia of biological inheritance. 51 years, not a loose estimate. Not a wide biological range that averages out to something meaningless. A specific, tightly clustered number controlled by approximately 290 genetic variants, identified in some of the most comprehensive genomic studies ever conducted on female reproductive biology. The heritability of menopause timing, the degree to which the age at which your fertility ends was inherited from your mother and will be inherited by your daughter, is approximately 44%. That is comparable to the heritability of height. Your menopause timing is nearly as genetically determined as how tall you are. Let that land for a moment. The end of your fertility is not something that simply happens to you the way your hair turns grey or your joints accumulate decades of use. It is a scheduled event. It runs on a timer that was set before you were born, governed by genes that were selected for, maintained and refined across the full span of Homo sapiens' existence. The oldest known fossils of our species were recovered from a site called Jebel Irhoud in Morocco and dated to approximately 300,000 years before the present. The women whose bones those fossils preserve carried the same genomic menopause architecture you carry. 300,000 years, 120,000 human generations, and in every generation of women across that span, the transition arrived on approximately the same schedule, steered by the same genetic machinery, toward the same biological destination. The question the evidence in this script builds toward answering is not whether this transition is designed. That question is settled. The genetic precision alone answers it. The question is why. Why did selection build the permanent end of female fertility into the human female body at 51, rather than allowing fertility to continue, as it does in virtually every other mammal on Earth, until near the end of life? What evolutionary calculation was so compelling, so repeatedly verified by the outcomes it produced across 300,000 years of selection, that the genomic architecture enforcing it was maintained with the same fidelity as the architecture governing height, bone density, and cardiovascular function? And here is the part that most people have never been told. The answer is not small. It is not a footnote in evolutionary biology. It is, arguably, the specific mechanism through which the human species developed the cooperative intelligence, intergenerational knowledge transmission, and social organizational capacity that separates us from every other primate on the planet. The permanent end of female fertility at 50 was not a concession to the aging ovary. It was the prerequisite for the most important social function the species produces. The non-adaptive view of menopause, the position that it is simply what happens when a woman outlives her eggs, a biological accident of extended human lifespan with no particular evolutionary purpose, faces a problem that researchers across multiple disciplines have been working to articulate for decades. The problem is 5 species of ocean-dwelling mammals who have absolutely nothing to do with modern medicine, extended human lifespan, or the cultural constructions of industrialized societies, and who nevertheless experience menopause with a biological completeness and a post-reproductive lifespan duration that makes the adaptive argument impossible to dismiss. In 2024, a landmark study published in Nature examined lifespan data from 32 species of toothed whales. The study identified 5 species in which females experience pronounced menopause: killer whales, short-finned pilot whales, false killer whales, narwhals, and belugas. These 5 species, combined with humans, are the only mammals on Earth known to experience the permanent cessation of fertility, followed by a substantial post-reproductive lifespan. The study's central finding was this: females of the 5 menopausal whale species live, on average, approximately 40 years longer than females of non-menopausal whale species of comparable body size. Not slightly longer, not marginally extended, an entire additional lifespan. And the mechanism was clear in the data. Menopause in these species evolved not by shortening the reproductive phase. The end of fertility arrived at approximately the same biological age as in non-menopausal relatives, but by extending total lifespan outward into the post-reproductive direction. The body kept running, the fertility stopped, something else began. What began is worth understanding precisely, because it is the same thing that begins in you. Killer whales and humans share no recent common ancestor. They are separated by more than 90 million years of independent evolution, entirely different physiologies, entirely different environments, entirely different genomic histories. They did not inherit menopause from a shared ancestral lineage. They each arrived at the same solution independently through convergent evolution, driven by the same underlying adaptive logic. When 2 lineages separated by 90 million years independently evolve the same biological structure to solve the same problem, the word scientists use for that convergence is not coincidence, it is evidence. Evidence that the problem is real, the solution works, and the evolutionary pressure that selected for the solution was operating in both lineages with sufficient consistency and force that 2 entirely different biological architectures reach the same answer across 9 decades of millions of years of independent development. The problem both species solved is this: in highly social species with long intergenerational contact and complex cooperative rearing, experienced post-reproductive females provide a form of group survival infrastructure so valuable in food provisioning, social coordination, birth attendance, ecological knowledge transmission, conflict mediation, that their continued survival and active contribution to the group is worth more in terms of genes transmitted to future generations than the additional offspring an older fertile female could theoretically produce. This is the core of the adaptive menopause argument. And the whale evidence published in Nature and now constituting the most direct empirical test of adaptive menopause theory ever conducted, supports it with the kind of quantitative precision that moves an argument from interesting hypothesis to settled science. Across 10 chapters, the evidence assembles itself from multiple independent directions. The genomics of menopause timing, the whale comparative data, the forager field studies in Tanzania, the Philippines, Paraguay, and the Kalahari. The brain imaging research documenting what the menopausal transition is actually building in the neurological architecture of the post-reproductive female. The reproductive conflict data revealing the specific social dynamics that menopause resolves and what the resolution creates space for. And the 26 years, the specific number, confirmed in hunter-gatherer demographic data, representing the average post-reproductive lifespan that human women experience in forager societies, untouched by modern medicine, through which to understand what the design was always intended to produce. You're not at the end of something. The evidence is unequivocal on this point. You're at the beginning of the stage of your biological life that took 300,000 years to engineer, toward which your entire previous reproductive life was building, and for which your body has been preparing with a structural depth and neurological specificity that no medical conversation has yet adequately described to you. Let the first chapter begin to show you what that preparation looks like from the inside. Every other mammal on Earth remains fertile until death or near death. This is not a generalization that softens at the edges when examined closely. It is the empirical baseline of reproductive biology across more than 5,400 described mammalian species, from mice to elephants, from river otters to lowland gorillas. Reproductive senescence in mammals is gradual, corresponding approximately with somatic senescence, the body's general aging, meaning that fertility declines slowly over time, and the endpoint of fertility and the endpoint of life are close enough in time that no meaningful post-reproductive lifespan exists. The female chimpanzee, our closest living genetic relative sharing approximately 98.7% of our functional DNA, remains capable of conception until the final years of her natural lifespan. Studies of wild chimpanzee populations in Uganda and Tanzania document reproductive activity in females well past their mid-40s, with somatic and reproductive aging proceeding in parallel. Fertility ends when life ends. The chimpanzee does not schedule its ending earlier. The human situation is categorically different and demands a categorical explanation. The average human female undergoes the permanent cessation of fertility at approximately age 51. Population studies spanning European, North American, African, Asian and indigenous populations confirm this average with a consistency that signals tight genetic control rather than loose biological drift. Following this transition, a human woman in a forager society operating without modern medicine can expect to live approximately 26 more years, confirmed through demographic analyses of populations including the Hadza of northern Tanzania, the Kungsan of the Kalahari, and the Ashe of Paraguay, 26 years, a post-reproductive lifespan that, measured alone, exceeds the entire lifespan of the vast majority of mammalian species that exist. The human female does not age out of fertility gradually. She steps across a precisely programmed threshold while her body continues operating at full physiological capacity for decades afterward. The paradox is exact. Natural selection favours traits that increase reproductive output. A female who remains fertile longer leaves more descendants than a female whose fertility ends at 50. More descendants means more copies of her genes propagating forward through time. More copies means stronger selection for the traits that produce those copies. The basic mechanics of natural selection should have eliminated the early fertility end date from the human lineage within dozens of generations of its first appearance, unless the early fertility end date was producing something that more than compensated for the lost reproduction. Not slightly compensated, substantially overcompensated. Because the genetic architecture enforcing menopause timing has not merely survived selection, it has been refined under selection, sharpened, made more precise. The genomic evidence for this refinement is recent, specific, and difficult to argue with. Research published in 2024 in journals including Nature and Science Daily identified 4 genes with the largest known effects on menopause timing, finding that these genes are also significantly associated with cancer risk pathways, confirming that the genes controlling when the ovarian reserve depletes are not peripheral cellular housekeeping genes, but central regulatory genes embedded in the most fundamental machinery of cellular integrity. The mismatch repair gene MSH6, a gene central to DNA error correction processes that prevent cellular dysfunction, was identified in a previous genome-wide association study as significantly associated with menopause timing, with a p-value of 1.9 times 10 to the power of -9. A number that size in genomic research is not a marginal signal. Key, it is a definitive biological marker of functional importance. What this means in plain terms is that the genes governing when fertility ends are not isolated reproductive genes running their own separate program. They are genes woven into the fabric of fundamental cellular regulation, the same genetic networks that govern cell cycle control, DNA repair fidelity, and long-term somatic maintenance. Selection has bound menopause timing to the deepest levels of cellular biology, which is what selection does with traits it cannot afford to leave to chance. The menopause countdown is running on the most reliable genomic hardware the body possesses. Track the ovarian reserve across the female lifespan, and the precision of the schedule becomes visible in concrete numbers. A female fetus carries approximately 1 to 2 million potential eggs, ovarian follicles, at the time of birth. This number is fixed before birth. Unlike any other cell type in the body, it does not regenerate, it does not replenish. The entire lifetime supply is present at the beginning and decreases continuously thereafter, through a combination of monthly ovulation and the parallel process called follicular atresia, in which follicles die without ever being released. By puberty, the reserve has declined to between 300,000 and 400,000 follicles. By age 40, approximately 25,000 remain. At the onset of menopause, the count has fallen to fewer than 1,000. a threshold that the hypothalamus detects through the hormonal feedback loop and responds to by halting the production of the hormones that drive ovulation. The machinery does not malfunction at this point. It reads the signal it was always going to read and does precisely what it was always going to do when it read it. But here is where the paradox sharpens rather than dissolves. The depletion schedule itself, the rate at which follicular atresia removes eggs across the decades between birth and menopause, is not simply a passive biological drain. It is governed by those 290 genetic variants, by those DNA repair genes, by the same MSH6 and its molecular companions, all of which control how rapidly the ovarian reserve responds to cellular stress signals and initiates the programmed death of follicles. The rate of depletion is programmed. The endpoint at approximately 1,000 follicles remaining is programmed. The age of approximately 51 at which this endpoint is reached, given a starting reserve of 1 to 2 million and a depletion rate governed by the full ensemble of these genetic variants, is programmed. 51 is not where the eggs happen to run out. 51 is where the design calculated the transition should occur. The calculator ran for approximately 300,000 years of human evolution, with every generation providing a data point about which menopause timing produced the most surviving descendants, and the answer it converged on is 51. Why 51 rather than 45? Why not 58? The answer requires understanding not just the biology of the ovarian reserve, but the specific relationship between a woman's accumulated social and ecological knowledge at different ages and the value of that knowledge to the group she lives within. It requires understanding something that the next chapter begins to address through the most unexpected comparative evidence in the entire menopause literature. Evidence drawn not from any human population, but from the open ocean, where 5 species of toothed whales arrived independently at the same biological answer that human evolution produced, and where the data on what post-reproductive females contribute to group survival is so precise and so quantitatively dramatic that it removes ambiguity from the argument in a way that human data, always messier and more culturally complicated, cannot fully achieve on its own. The question planted here, why 51, why that specific coordinate, does not close in this chapter. It will close only after the whale evidence has been examined, after the forager field data has been presented, and after the neurological evidence from brain imaging research has revealed what the female brain is actually doing in the years surrounding the transition. But the question itself is already doing its work, because 51, understood as a deliberately programmed coordinate rather than an accidental endpoint, changes every single thing about how you understand the decades that follow it. Place this number against the scale of mammalian biology and let it register properly. 5,400. That is the approximate number of currently described mammalian species, from the bumblebee bat weighing 2 grams to the blue whale exceeding 150 metric tons, from the naked mole rat whose biology has fascinated cancer researchers for decades, to the African elephant whose memory and social complexity has generated a substantial scientific literature of its own. 5,400 species, across all of them, across every variation in body size, lifespan, reproductive strategy, social structure, and ecological niche that mammalian evolution has produced across 200 million years of diversification. Exactly 6 are known to experience menopause. The cessation of fertility followed by a substantial post-reproductive lifespan is one of the rarest biological phenomena in the entire class Mammalia. 6 out of 5,400 is not a sample size, it is a signal. When a biological phenomenon appears at a frequency of 0.11%, across a taxonomic class, its distribution is not random. Something specific, some precise and unusual convergence of ecological, life history and social conditions, is required before evolution will build this particular design, and understanding what those 6 species share is the most direct possible route to understanding why menopause exists at all. The 6 are humans, killer whales, short-finned pilot whales, false killer whales, narwhals, and belugas. 1 primate and 5 toothed whales, cetaceans, the group that returned to the ocean from terrestrial ancestors approximately 50 million years ago and developed, in the process, some of the most sophisticated social brains and cooperative behavioral systems of any non-human mammals alive. The fact that all 5 non-human menopausal species are toothed whales is not a coincidence produced by phylogenetic proximity. These 5 species are not each other's closest relatives within the cetacean order. They are distributed across the toothed whale lineage in a pattern that indicates menopause evolved independently multiple times within the group, as it also evolved independently in the human lineage, driven by the same adaptive logic operating in different genomic architectures toward the same functional solution. The 2024 Nature study from the University of York and the University of Exeter is the most comprehensive quantitative treatment of this rarity. The research team compiled lifespan, reproductive, and demographic data for 32 species of toothed whales, comparing the 5 menopausal species against 27 non-menopausal relatives. The central measurement was post-reproductive lifespan, the number of years females in each species continue living after their final reproduction. In the 5 menopausal species, this number is dramatic. Killer whale females stop reproducing in their 30s and 40s and live into their 60s, 70s, and occasionally their 90s, a post-reproductive lifespan routinely exceeding 40 years. Short-finned pilot whale females, documented in long-term studies off the Canary Islands, continue living for decades after their final calf. The pattern holds across all 5 menopausal species. Fertility ends at a specific age. Life continues at full physiological capacity for a substantial fraction of the total lifespan. In the 27 non-menopausal whale species, post-reproductive lifespans are essentially 0. Females continue reproducing until near death. Reproductive and somatic aging track each other. The endpoint of fertility and the endpoint of life coincide. The magnitude of the difference that menopause makes to female lifespan is the 2024 study's most important quantitative finding. Females of the 5 menopausal whale species live approximately 40 years longer than females of non-menopausal whale species of comparable body size. Not marginally longer, not somewhat longer in certain favourable environmental conditions. Approximately 40 years longer as a consistent biological pattern across the 5 species and their decades of collected life history data. And crucially, and this is the finding that eliminates the most common alternative explanation, this extended lifespan was produced not by a shorter reproductive phase but by an expanded post-reproductive phase. The reproductive period in menopausal whales is not shorter than in non-menopausal relatives. Fertility ended at approximately the same biological age. What changed was that the body kept running for 40 more years after fertility ended. Personal DNA connection. Bomb 01, your body is doing the same thing. Not as a cultural response to modern medicine. Not as a byproduct of improved nutrition. As a biological program running the same architecture that killer whales run, built by the same evolutionary logic toward the same functional destination. A post-reproductive female whose continued active life within her social group produces more surviving descendants through the survival of her children's children and the social infrastructure she maintains for her entire community, than any additional offspring she could theoretically produce herself. The specific conditions that appear consistently across all 6 menopausal species are worth enumerating carefully because they define the evolutionary prerequisites. All 6 species are long-lived, with lifespans substantially exceeding the mammalian average for their body size, allowing for the development of genuinely long intergenerational contact between older females and the younger generations whose survival they support. All 6 are highly social, living in stable, complex social groups where individual relationships persist across years and decades, where accumulated social knowledge has operational value, and where the loss of a single experienced individual is a group-level event with measurable demographic consequences. All 6 engage in extensive cooperative rearing, with investment in infant survival coming from multiple group members, rather than being concentrated in the biological mother alone. And all 6 have ecological conditions under which accumulated environmental knowledge of food sources, migration routes, seasonal resource patterns, danger signals, provides a survival advantage that increases with the age of the knowledge holder. These are precisely the conditions under which experienced older females become the group's most critical survival infrastructure. Not the most physically powerful members, not the most fertile members, the most experienced members, the ones who have navigated the environment long enough to carry its history in their bodies, who have attended enough births and crises and resource shortfalls to know what the current situation resembles and what responses have worked before, and who are past the period of their own intensive reproductive investment and therefore fully available to direct their biological resources toward the group's future rather than their own immediate reproductive success. In all 6 species, this is what postmenopausal females do, and in all 6 species, the data shows that when they cannot do it, when they are absent or die, the consequences for the group are measurable, specific and significant. The full depth of those consequences is what the following chapter begins to examine, starting with the species whose long-term data is the most precise and whose postmenopausal female contributions have been tracked with enough quantitative rigour to move the argument from inference to evidence. The question left open at the close of the previous chapter was pointed. When postmenopausal females are absent from their groups, what specifically happens? The answer exists in the data, in killer whale populations, and the data is more dramatic than almost anyone who has not read the primary research expects it to be. Long-term research on killer whale populations in the Pacific Northwest, conducted over multiple decades by the Centre for Whale Research and associated institutions, has accumulated individual-level demographic data of a quality that few wildlife studies achieve. Researchers can identify individual killer whales by the distinctive saddle patches behind their dorsal fins, track them across years and decades, document their reproductive histories, record their deaths, and measure the demographic consequences of their loss for the surviving members of their social groups. This data has been used to test, with a precision that ethnographic human data rarely permits, the specific contribution of post-reproductive females to the survival of younger group members. The findings are specific and sobering. In killer whale pods, post-reproductive females serve as the primary ecological knowledge holders for their social group. Research published in Proceedings of the Royal Society B, examining data from 102 killer whales tracked across multiple years, found that post-reproductive females lead their groups to food sources at disproportionately high rates, and that this leadership is most pronounced in years of environmental scarcity. When Chinook salmon, the killer whale's primary prey in Pacific Northwest waters, are at low abundance, in good years, when food is plentiful, the leadership advantage of postmenopausal females is detectable but modest. In poor years, when finding food requires navigating through depleted salmon runs using knowledge of where abundance has historically recovered, their leadership advantage becomes the difference between adequate nutrition and caloric deficit for every member of the pod. The interpretation is direct. Post-reproductive females carry the group's deepest ecological memory. They have experienced the most cycles of abundance and scarcity. They know which foraging areas hold through poor salmon years because they have watched those areas hold through previous poor years. The younger females in the group, however physically prime and however reproductively active, cannot replicate this knowledge. It is not genetically transmitted, it is experientially accumulated, and the only way to acquire 40 years of ecological memory is to have been alive and paying attention for 40 years. Now, the mortality data. When a post-reproductive female killer whale dies, what happens to the members of her social group? The data shows mortality consequences that are remarkable in their magnitude. Research examining multi-generational killer whale demographic records found that in the year following the death of a post-reproductive female, the mortality rate of her adult sons increased by a factor of approximately 14 compared with periods when she was alive. Adult sons, not dependent calves. Fully grown adult males who have been behaviourally independent for years. The loss of their post-reproductive mother increases their probability of dying within 1 year by 14 times. For daughters, the mortality increase following the loss of a post-reproductive mother is also statistically significant, though less extreme than for sons. The difference between the son effect and the daughter effect reflects a specific feature of killer whale social structure. Males remain in their natal pods throughout their lives, maintaining continuous close association with their mothers, while females sometimes transfer between pods. The son's survival is more tightly coupled to their post-reproductive mother's continued presence because the relationship is permanent, and when that permanent presence ends, the sons, even fully adult, behaviourally independent prime condition males, show a mortality spike that is difficult to explain through any mechanism other than the loss of the experienced female whose ecological knowledge they had been relying on, and whose social mediation within the pod structure they had been benefiting from, without fully accounting for the benefit. This is the revelation that makes the whale evidence more than an interesting biological comparison. It demonstrates that post-reproductive females are not passive beneficiaries of the group's tolerance for their continued presence. They are active, indispensable, specifically irreplaceable contributors to the survival of group members who are themselves in the prime of their biological capacity. The post-reproductive killer whale female is not maintained in the group out of social sentiment. She is maintained because removing her produces a 14-fold mortality spike in adult males. Selection maintained her because groups that kept her alive had more surviving members and therefore more surviving descendants than groups that did not. The parallel to human populations is not metaphorical, it is structural. The specific conditions that make postmenopausal killer whale females irreplaceable, accumulated ecological knowledge, social coordination capacity, multi-generational relationship networks, absence of competing reproductive demands, are the same conditions that make postmenopausal human females irreplaceable in the forager contexts that constitute the entirety of human evolutionary history before approximately 10,000 years ago. Yet the killer whale data quantifies something that the human ethnographic data has been indicating qualitatively for decades, but that the cultural complexity of human societies and the methodological challenges of studying them have made difficult to measure with equivalent precision. The whale evidence closes the first secondary mystery. Only 6 mammal species experience menopause because only 6 species have independently developed the specific convergent combination of long lifespan, complex sociality, cooperative rearing, and experiential ecological knowledge transmission that makes post-reproductive female expertise worth the evolutionary cost of building it. The 5 toothed whale species in humans are the 6 species that met all 4 conditions in 2 entirely independent evolutionary lineages, and arrived at the same answer: stop the fertility, keep the female. The evidence for what she does in the years after fertility ends is too consistent, too quantitatively significant, and too perfectly aligned across species to permit any other conclusion. But the whale evidence, as complete as it is, does not explain the mechanism in humans. It does not show what human grandmothers specifically do that produces survival advantages for their grandchildren and the broader social group. That evidence comes from a very different source: decades of field research in the Tanzanian savannah, watching specific women dig for specific tubers in a specific dry season landscape and measuring the growth curves of the children those women fed. The next chapter builds that case from the ground up with the numbers that make it concrete. Stand at the edge of a dry woodland in what is now northern Tanzania. The time is not this time. The landscape is the Hadza homeland, a mosaic of acacia scrub, seasonal grassland, and rocky escarpment surrounding the eastern shore of Lake Eyasi, a landscape that anthropologists from the University of Utah have been visiting since the 1980s to study one of the last remaining hunter-gatherer populations on Earth, whose subsistence patterns offer a window into the foraging ecology of our evolutionary past. The morning air is approximately 18 degrees Celsius. The dry season has been running for 2 months. The wet season food sources, berries, honey, above-ground vegetation, are reduced or absent. What remains is underground. Tubers, the starchy, fibrous storage organs of plants that invest their calories below the surface specifically because above-ground exposure would cost them to herbivores. Finding them requires knowing where to look. Digging them requires knowing how deep to go. Knowing both requires having done it in this specific landscape, in this specific dry season condition, for years. The woman working 30 meters from the tree line is not the youngest woman in this forager camp. She is past the age at which the camp's younger women will have children. Her hair carries the grey of decades. Her hands carry the specific muscle development of sustained habitual tool use, enlarged attachment points at the metacarpals, worn palmar surfaces that have been building callus for 30 years of daily digging. She works without apparent effort in the sense that every movement is economical, no wasted action. She finds the plant she's looking for by reading surface signs that a younger woman would walk past without seeing. A specific leaf shape half hidden under dry grass, a subtle soil mound indicating a tuber at depth. The particular spacing between plants that tells her the root system she's targeting runs in a direction that makes digging from the eastern side more productive than from the west. She has been doing this since she was approximately 20 years old. She's now 55. That is 35 years of this specific landscape, in every dry season and wet season that landscape has produced in her lifetime. Her internal map of this ecosystem is the most comprehensive single cognitive resource the group possesses for the question of where food is when the preferred food is absent. Kristen Hawkes at the University of Utah understood what she was looking at when she first observed this pattern in the Hadza data collected across field seasons in the 1980s. The data showed something that did not fit the then-dominant model of human evolution, a model focused on male hunting as the primary driver of the species' social and cognitive development. The Hadza data showed that the most productive foragers in the population, measured in calories acquired per hour of work, were not the young males engaged in hunting. They were the post-menopausal women engaged in tuber digging, and the calories those women produced were not going to themselves. The data showed a specific correlation between how much post-menopausal women foraged and how well the weaned children of younger women in the same camp grew. The correlation was not marginal. When Hawkes and colleagues analysed the relationship between Hadza children's weight gain and the foraging effort of the women around them, they found that the single strongest predictor of a child's weight gain during the critical weaning period, after the child's mother stopped nursing but before the child could efficiently forage for itself, was the foraging effort of the child's maternal grandmother, not the mother's foraging effort, because the mother's foraging was limited by the demands of the next pregnancy or infant, not the father's hunting output. Because hunting success was too variable and too infrequent to provide the reliable daily caloric base that a growing child requires. The grandmother's daily tuber production was the most statistically reliable predictor of whether a specific child gained or lost weight in the weeks and months following weaning. Evidence block. 4 specific data points make this concrete. First, Hadza postmenopausal women spend more hours per day in active food acquisition than any other demographic group in the population, exceeding the foraging hours of reproductive age women and reproductive age men in the data collected across multiple dry season and wet season field periods. Second, the primary food source acquired through this foraging effort is underground tubers, which require accumulated landscape knowledge to locate efficiently, and which represent the most reliable caloric source during dry season periods when surface foods are depleted. Third, the proportion of this food production that enters the grandmother's own caloric budget is substantially smaller than the proportion directed toward the nutritional support of her daughter's young children, a pattern consistent with allomaternal provisioning rather than self-maintenance foraging. Fourth, in Agta forager communities in the northern Philippines, researchers found that the average interbirth interval, the time between successive births by a mother, was approximately 2.7 years across 258 births recorded in the demographic data. Critically, grandmother presence was associated with shortened interbirth intervals in the communities where this relationship was examined, meaning that when a grandmother was present and actively foraging for grandchildren, the mother could wean her current infant earlier and become pregnant again sooner, increasing her total lifetime reproductive output. This last data point is where the grandmother hypothesis becomes mathematically powerful. The argument is not simply that grandmothers improve child survival by providing food. It is that grandmothers enable mothers to have more children in a lifetime by reducing the interbirth interval, and that the cumulative effect of this enabled fertility, across multiple daughters and multiple generations, produces more grandchildren and great-grandchildren for the grandmother than she could have produced by remaining fertile and continuing to bear her own children. A postmenopausal grandmother who enables her 2 daughters to each have 1 additional child by shortening their interbirth intervals through active food provision has contributed the equivalent of 2 additional births to the family line, without the mortality risk of late pregnancy, without the metabolic demands of late-stage maternal investment, and without the competitive resource conflict that would arise if she were simultaneously pregnant alongside her daughters. This is the grandmother's calculation, not a conscious calculation. A biological one, running in the background of natural selection's bookkeeping across thousands of generations of Pleistocene forager life. The calculation is, at what age does a woman's contribution to the transmission of her genes through her grandchildren's survival exceed her contribution through her own fertility? The answer, refined across 300,000 years of selection in the specific ecological and social conditions of forager life, is approximately 50. Not because 50 is a round number, because 50 is the specific inflection point at which accumulated knowledge and available investment capacity combine in the post-menopausal female body to make her more reproductively effective as a grandmother than as a mother. Biological controversy. Bomb 01. The grandmother hypothesis is not universally accepted as a sufficient explanation for the evolution of human menopause, and the challenges to it are serious enough to deserve direct examination. One significant critique is that the hypothesis struggles to explain the initial evolution of menopause, the problem of how the first females with early fertility endings could have benefited genetically from a trait whose benefits were tied to the existence of already present grandchildren and already established cooperative social networks. A trait that requires the pre-existence of the very social structures it is supposed to explain faces a chicken and egg problem that the hypothesis has not fully resolved. A second challenge is that grandmothering effects, while present in the Hadza and several other study populations, are not consistently strong across all forager societies examined. The AGTA data shows weaker grandmother effects than the Hadza data, and some populations show effects that are demographically positive, but not of a magnitude obviously sufficient to drive the evolution of a 26-year post-reproductive lifespan. A third challenge comes from the observation that male relatives, specifically maternal grandfathers and paternal grandparents, also contribute to child survival in some populations, suggesting that extended lifespan parental investment is not uniquely female, even if the pattern is consistently stronger on the maternal grandmother side. Scientific Debate Invitation 01. These challenges do not invalidate the grandmother hypothesis, they refine and complicate it. The most current synthesis in the field treats grandmothering not as the sole explanation for menopause, but as the primary driver operating alongside a second explanatory framework, the reproductive conflict hypothesis, that addresses the chicken and egg problem by explaining why menopause would have been initially selected for in specific social circumstances even before its grandmothering benefits were fully operational. That second framework is the subject of a later chapter. For now, the evidence from Hadza field data, from AGTA demographics, from the Otkung-san and Ashe populations, and from the comparative whale literature converges on a conclusion that the data does not permit easy dismissal. Postmenopausal female investment in grandchildren survival is a consistent cross-cultural, cross-species pattern that has measurable demographic consequences and that is calibrated precisely to the biological capacities of women in their post-reproductive years. The grandmother's calculation is real. It runs in living forager populations today, and it was running in exactly the same way in the landscape of Pleistocene Africa 300,000 years ago, in the bodies of the women whose bones are the oldest known Homo sapiens fossils in the world. The evidence for adaptive menopause does not rest on any single line of inquiry. It accumulates from multiple independent directions, each carrying its own methodological limitations and its own specific evidentiary strengths, and the convergence point where all lines meet is where the scientific case is strongest. Begin with the phylogenetic evidence. In 2024, the Nature study examining toothed whale lifespan data made a finding about the evolution of post-reproductive lifespan that resolves a long-standing debate within the menopause literature. Menopause evolved by extending total female lifespan, not by reducing the reproductive lifespan. This is the critical distinction. The non-adaptive hypothesis of menopause predicts that postmenopausal lifespan in humans is a byproduct of extended somatic lifespan, that the body started living longer due to improved nutrition, medicine, or other factors, while the reproductive period remained unchanged, producing a post-reproductive gap as a passive consequence. The adaptive hypothesis predicts something different, that selection specifically extended female lifespan beyond the reproductive period, creating the post-reproductive phase actively, rather than passively allowing it as a side effect. The whale data settles the question by showing that in the 5 menopausal species, the reproductive period is not shorter than in comparably sized non-menopausal relatives. The fertility end date is approximately the same. What is different is that these females keep living for 40 additional years after fertility ends. Selection extended the post-reproductive period. It did not shorten the reproductive one. For humans, the genetics support the same conclusion. The 290 genetic variants controlling menopause timing do not merely determine when the ovarian reserve runs out. They are part of a broader genomic architecture controlling the relationship between reproductive senescence and somatic senescence, the divergence between how quickly the ovaries age and how quickly the rest of the body ages. In human females, this divergence is extreme by mammalian standards. Ovarian function declines and stops at approximately age 51, while somatic function, cardiovascular health, cognitive function, musculoskeletal capacity, immune competence, continues at levels adequate for several additional decades of active foraging, social coordination, and birth attendance. Selection has specifically engineered the female body to outlast its own ovaries by a meaningful margin. The ovaries were not designed to last a lifetime. They were designed to last until 50. Everything else was designed to last longer. Now the forager demographic evidence in its full convergent form. Population-level data from 4 forager societies, the Hadza of Tanzania, the Kungsan of the Kalahari, the Aceh of Paraguay, and the Agta of the Philippines, shows a consistent pattern that transcends cultural, geographic, and ecological variation. In all 4 populations, postmenopausal women invest more time per day in food acquisition for dependent young than any other age-sex demographic category. In all 4 populations, the relationship between grandmother foraging effort and grandchild nutritional status is statistically positive, meaning children with actively foraging grandmothers do better nutritionally than children without them. In all 4 populations, grandmother presence is associated with shorter maternal interbirth intervals, meaning mothers with active grandmothers available can wean earlier and reproduce again sooner, increasing their lifetime reproductive output. The specifics vary across populations, the magnitude of the effect, the primary food sources involved, the degree to which the grandmother effect is mediated through maternal versus paternal lines. But the directionality is invariant. Grandmother presence helps, grandmother absence costs. Personal DNA connection, BOM02. The invariance across 4 culturally distinct populations practicing subsistence strategies as different from each other as the Tanzanian savanna is from the Philippine forest is the strongest possible indicator that this pattern is not culturally constructed. It is biologically generated. Your own postmenopausal foraging drive, the motivation toward productivity, toward practical contribution, toward ensuring that the people you care about have what they need, is not a personality trait. It is a biological program running in your body with the same consistency it runs in the body of a Hadza grandmother digging tubers in a dry season landscape, and with the same underlying purpose, the genomic calculation that your contribution through your grandchildren's survival is more effective in terms of genes transmitted to future generations than your contribution through your own additional fertility would be. Now the reproductive conflict evidence, which provides the second explanatory axis that the grandmother hypothesis alone leaves unresolved. In 2008, evolutionary biologists Michael Kant and Rufus Johnstone published a hypothesis in the journal Evolution and Human Behavior, proposing that menopause evolved not only because of its grandmothering benefits, but because of the costs of intergenerational reproductive conflict that it eliminates. The argument is elegant. In human societies, and particularly in the patriarchal societies common in hunter-gatherer contexts, where women move to live with their husbands' families after marriage, older women and younger women of the same social group face a specific competitive dynamic. An older woman whose son has brought home a mate, or whose daughters-in-law are now bearing children, is competing with those younger women for the same finite pool of resources, food, male provisioning attention, social support from the group, that they are competing for. Fertility overlap between generations creates a resource conflict. Menopause removes the older woman from fertility competition precisely when that competition would be most costly, when her accumulated knowledge and social experience could be most effectively directed toward cooperation rather than competition. Field data from the Bostock Kungsan and the Hadza, examined to test the reproductive conflict hypothesis, found that in groups where older and younger women's reproductive periods did overlap, the cooperative provisioning that characterizes the grandmothering relationship was reduced relative to groups where the older woman had already passed through menopause and was no longer a reproductive competitor. The removal of fertility competition created cooperative space. The cooperative space allowed the grandmothering investment to operate at full effectiveness. Menopause, in this reading, was not just selected for its positive benefits, the grandmothering contribution, but also for the negative benefits it provided by eliminating the reproductive competition that would have diminished cooperative relationships and reduced the social cohesion of small forager groups depending on those relationships for survival. Secondary mystery 01 closes here. Only 6 mammal species experience menopause because menopause requires a specific and unusual combination of adaptive conditions: long lifespan enabling intergenerational contact, complex sociality making post-reproductive female expertise valuable, cooperative rearing making older female investment operationally effective, and either patriarchal social structure or equivalent intergenerational reproductive competition that menopause specifically resolves. Humans and five-toothed whale species independently developed all of these conditions, independently ran the evolutionary selection process that the conditions create, and independently arrived at the same answer: design the female body to stop reproducing at a specific age and keep functioning for decades afterward. The convergence across 90 million years of evolutionary independence is the most powerful empirical confirmation the adaptive hypothesis has ever received. But the convergence evidence answers only the question of whether menopause is designed. It does not fully answer the question of what the design is doing inside the body, during and after the transition. What is actually happening in the brain during the menopausal transition that makes it feel so profound, so disorienting, and so structurally significant? And why does the transition, which is building something rather than ending something, come with such a demanding physical and neurological price? That question, the second secondary mystery, opens in the following chapter with the brain imaging data that has changed how researchers understand the menopausal transition at the most fundamental neurological level. The symptoms of menopause are not subtle. Hot flashes arriving without warning, the core body temperature suddenly seeming to rise by several degrees while the hypothalamic thermostat attempts to correct a signal it is misreading, generating the vasodilation, flush and sweating response that the body uses to cool itself from genuine overheating. Sleep architecture fragmented by night sweats, by the same thermoregulatory miscalibration occurring in the hours when sleep should be deepest. Cognitive fog descending and lifting unpredictably. The specific frustrating experience of reaching for a word or a memory, and finding it temporarily inaccessible in a mind that was, before the transition, reliably precise. Mood variability that does not correspond to external circumstances with the consistency it showed before. Joint discomfort. Energy patterns that shift in ways that feel like loss, but that do not behave exactly like the fatigue of illness or the tiredness of overwork. These symptoms are real. The research confirms them. The biological mechanisms producing them are well characterized. Hot flashes result specifically from estrogen withdrawal acting on the KNDY neurons in the hypothalamic arcuate nucleus, neurons that regulate body temperature and that, accustomed to operating in the presence of estrogen's modulating signal, misread its absence as a genuine overheating event and trigger a cooling response to correct a temperature that is not actually elevated. The cognitive changes correspond to demonstrable alterations in glucose metabolism in specific brain regions, documented in neuroimaging studies examining brain function across the menopausal transition. The mood variability corresponds to measurable changes in serotonergic and dopaminergic signaling during the transition period. These are not imagined symptoms. They are the physiological signatures of a brain undergoing structural reorganization. The paradox is this: if menopause is a design rather than a deterioration, if the transition is activating something rather than ending something, why does the activation feel like this? Why does a biological program that has been maintained under selection for 300,000 years, because of the functional competence it produces in postmenopausal women, arrive with a transition cost that mimics, from the inside, the experience of physical and cognitive loss? The answer, and it is an answer that recent neuroimaging research has made increasingly specific, is that the symptoms are the construction costs of a significant renovation, not a minor adjustment. A structural rebuild of the brain's reward architecture, hormonal sensitivity configurations, connectivity patterns, and attentional allocation systems. The menopausal brain is not declining. It is being rebuilt for a different operational purpose, and the disruption of the construction phase is the price of the renovation rather than evidence that the renovation is failing. The neuroimaging data is specific. Research published by researchers at Weill Cornell Medicine in 2024 using brain scanning to measure estrogen receptor density across women at different stages of the menopausal transition, found a pattern that runs counter to the simple narrative of hormone decline leading to brain decline. Estrogen receptor density in several estrogen-regulated brain networks increased during the transition, not decreased. The brain was upregulating its sensitivity to the declining estrogen signal, compensating for reduced hormone availability by increasing receptor expression in the circuits that depend on estrogen modulation. This is not what a brain undergoing passive deterioration does. This is what a brain under adaptive pressure does, responding to a changing hormonal environment by reconfiguring its receptor architecture to maintain and then redirect the functions those receptors serve. The grey matter evidence shows the same pattern in a different register. Neuroimaging research has documented reductions in grey matter volume in specific brain regions during the menopausal transition, reductions that correspond to the cognitive and emotional symptoms the transition produces. But the same research, examining women at later stages of the postmenopausal period, finds something that the simple hormone loss narrative does not predict: partial recovery. Grey matter volume in some regions shows neuroplastic regeneration following the acute phase of the transition, with the postmenopausal brain demonstrating compensatory growth in specific areas, consistent with a restructuring toward different functional connectivity patterns rather than a simple progressive loss. Community location prompt 01. Somewhere right now, a woman is navigating the menopausal transition in the dark, not literally, though perhaps literally, without anyone having told her that the brain fog she's experiencing is construction dust, that the thermoregulatory chaos is the body burning old calibrations to make space for new ones, that the sleep disruption and the mood variability and the sense of profound internal reorganization she may be feeling are the symptoms of a 300,000-year-old design running exactly on schedule. If that woman is you, or if you know exactly who she is, leave something in the comments. A word, a city, a gesture of recognition. This narration is specifically for her. What is being built in the renovation? The evidence points toward a specific set of functional capacities that the postmenopausal brain expresses more fully than the premenopausal brain. The theory of mind processing, the neurological capacity to model the mental and emotional states of multiple other people simultaneously, shows measurable enhancement in postmenopausal women in studies using functional brain imaging during interpersonally complex tasks. The default mode network, the brain's primary social cognition infrastructure, active when the brain is processing social relationships, anticipating others' responses, and integrating long-term memory with present-moment social demands, shows altered and in some respects strengthened connectivity in postmenopausal women compared with premenopausal controls. Lace cortisol recovery following acute social stress, the speed with which the body returns to baseline physiological calm after an emotionally demanding social interaction, is measurably faster in postmenopausal women than in women in the most intensive phase of their reproductive years. These are not random improvements. They are a specific profile of enhanced social cognition, improved stress regulation, and deepened interpersonal processing capacity. Precisely the capacities required for the social coordination work that the grandmother hypothesis assigns to postmenopausal women: birth attendance, intergroup mediation, conflict resolution within small forager bands under resource stress. The coordination of food provisioning across multiple family units. The management of the social relationships that hold cooperative groups together across the periods of difficulty that periodically threaten their cohesion. The brain is being built for this work during the transition that feels like it is ending something. The heat, the sleeplessness, the cognitive reorganization. They are the price of a renovation whose product is a social brain more specifically suited for the most important coordination work the human species produces than any brain it possessed before the transition occurred. The reproductive conflict hypothesis begins with a mathematical reality that becomes a social problem. In any species where multiple generations of females can be simultaneously reproductively active and simultaneously dependent on the same finite pool of resources, competition between those generations is not a social failure. It is a biological inevitability. Resources, food, male provisioning, social attention, physical space, birthing support, are limited. When two women in the same social group are simultaneously pregnant or nursing infants, they are competing for those resources with the directness and intensity that has no parallel in any other social relationship the group contains. The competition is not hostile in any conscious sense. It is structural, embedded in the biology of resource allocation and the arithmetic of group survival under constraint. Establish scales of this problem across human forager demographic data. In a typical hunter-gatherer band of approximately 50 individuals, the reproductive age female population at any given time includes between 8 and 12 women. Of those, typically 2 to 4 are simultaneously in late pregnancy or early nursing, the periods of maximum caloric demand. The caloric deficit of late pregnancy is approximately 20 to 25% above basal metabolic rate, equivalent, in an active foraging woman covering 9 to 14 kilometers daily, to an additional 500 to 700 kilocalories per day. i.e., 2 women simultaneously in late pregnancy in a band of 50 people represent a combined additional caloric demand of approximately 1,000 to 1,400 kilocalories per day on a food supply that is already fully claimed by the group's nutritional requirements. The resource arithmetic is tight before conflict is added to it. When it is added, when the 2 women are mother-in-law and daughter-in-law in a patrilocal group where the older woman's son is the younger woman's mate, the social friction that the caloric competition generates has consequences for group cohesion that extend well beyond the nutritional arithmetic. Kant and Johnstone, in their 2008 paper presenting the reproductive conflict hypothesis, made an argument that the evolutionary literature had been circling for years without stating in fully formal terms. The argument is this. In species with patrilocal residence patterns, where females disperse to live with their mates' families, older females and their daughters-in-law share no genetic relationship. They are not relatives. Their genetic interests align only through their shared investment in the man connecting them, the older woman's son, the younger woman's mate. When both are reproducing simultaneously, their resource interests conflict directly, without the genetic cushion that kin-relatedness would provide. Natural selection, over many generations, favoured older women who reduced this conflict by stepping back from fertility competition at the point where stepping back increased the survival of their grandchildren more than it decreased the survival of their own additional offspring. Menopause is, in this framework, not just a grandmothering strategy, it is a conflict resolution strategy, the biological mechanism through which older women remove themselves from a competitive dynamic that was reducing the cooperative investment the group needed to function. Biological Controversy Bomb 02. The reproductive conflict hypothesis has generated substantial scientific debate since 2008, and the debate is genuinely unresolved in ways that make the controversy worth examining carefully. The primary challenge is empirical. Not all human societies are patrilocal. A significant proportion of documented human cultures practice matrilocal or bilocal residence patterns, and the reproductive conflict hypothesis predicts that menopause timing and the benefits of postmenopausal grandmothering should vary between patrilocal and non-patrilocal societies in ways that the data has not consistently confirmed. The hypothesis also faces the same origin problem that challenges the grandmother hypothesis, explaining how the first menopause-displaying females could have benefited from a trait whose cooperative payoffs depended on the prior existence of social structures that menopause itself was supposed to help create. Scientific debate. Invitation 02. The 2 frameworks, grandmother hypothesis and reproductive conflict hypothesis, are increasingly viewed in the research literature not as competing explanations but as complementary ones, operating through different mechanisms toward the same adaptive outcome. The grandmother hypothesis explains the positive benefits of postmenopausal female investment. The reproductive conflict hypothesis explains the specific social conditions under which ceasing reproduction becomes more advantageous than continuing it. Both are probably partially right. The evidence suggests that menopause, like most complex biological adaptations, was not produced by a single selection pressure, but by a convergence of selection pressures that operated simultaneously, reinforced each other, and collectively produced the precise genomic architecture, the 290 genetic variants, the DNA repair gene associations, the 44% heritability, that controls the transition's timing today. What the reproductive conflict data most clearly illuminates is the social function of menopause that is invisible in the grandmothering data alone, the function of creating cooperative space within social groups by removing intergenerational reproductive competition. Research examining the patterns of food sharing, cooperative childcare, and social alliance formation in forager groups found that the cooperative relationships between older and younger women in these groups, the relationships through which grandmothering investment flows, are significantly stronger in groups where the older woman is postmenopausal than in groups where reproductive overlap between generations is present. The absence of fertility competition is not merely the absence of a negative. It is the creation of a positive, a cooperative relational capacity between generations that menopause specifically enables, and that no other biological mechanism in the human social repertoire produces with equivalent reliability. The full picture is beginning to emerge. Menopause at 50 is not the ending of the female biological contribution to the species. It is the scheduled resolution of the competitive conditions that limited that contribution throughout the reproductive years and the simultaneous activation of the post-reproductive biological capacities, neurological, hormonal, social, ecological, that allow the female body to serve its most sophisticated evolutionary function. The next chapter examines that function in the specific detail it deserves, what postmenopausal women do in their communities, in their families, and in the broader social networks of human survival that no census ever counted and no historical record ever adequately preserved. The unresolved question from the whale chapter was specific. Postmenopausal killer whale females, when they die, produce a 14-fold increase in adult male offspring mortality in the following year. What does that mortality spike tell us about what they were doing that prevented it? The answer involves two mechanisms, and both mechanisms have direct human parallels that the forager literature documents with enough specificity to make the translation from ocean to savanna more than analogical. The first mechanism is ecological knowledge transmission. In killer whale pods, post-reproductive females are the individuals most likely to lead the group to food sources during periods of scarcity. And long-term behavioral observation data shows that younger pod members follow and learn from these foraging decisions over years of observation and participation. When the knowledge holder dies, the pod does not immediately lose its foraging capacity, but it loses the specific ability to navigate novel scarcity situations that fall outside the experience of younger pod members, but that the post-reproductive female has encountered before, in a previous cycle of low salmon abundance and navigated successfully. The knowledge that died with her was the knowledge of what to do when conditions deviate from the current norm in a direction that younger individuals have not yet personally experienced. In human forager populations, this function is documented across every studied society that has multi-generational demographic data. Post-menopausal women in Hadza, Kung, Aceh, and Agta. Communities serve as the primary living repositories of environmental knowledge that has accumulated across decades of direct ecological experience. Knowledge of plant distribution, seasonal resource availability, water source reliability through drought, and the specific landscape signals that indicate conditions relevant to food acquisition decisions. This knowledge is not transmitted through language alone. It is transmitted through the shared practice of working together in the landscape. A younger woman foraging alongside an older woman for years, absorbing by proximity and participation the accumulated ecological reading of a 30-year relationship with a specific terrain. When the older woman dies, the transmission stops, and the younger woman, however capable, has not yet accumulated enough independent experience to replicate what was lost. The revelation at the midpoint of this chapter is the finding that makes this function more than foraging assistance. Postmenopausal women in forager societies are the primary intergenerational memory systems for social knowledge as well as ecological knowledge. The living archives of the group's history of relationship decisions, alliance formations, conflict resolutions, and demographic events. Research examining the social network structure of Hadza Nikung communities found that postmenopausal women occupy the most central positions in the inter-household social networks that organise food sharing, mutual aid, and cooperative childcare. They are not peripheral recipients of the group's cooperative capacity. They are the nodes through which that cooperative capacity is organised and sustained. The group's cooperative behaviour patterns flow through the relational networks that postmenopausal women maintain, and the structure of those networks, the specific patterns of alliance, obligation, and reciprocal support that make the group function as a unit rather than as a collection of competing family interests, is largely the product of the postmenopausal women's relationship-tending work. This work is almost entirely invisible in historical records, in archaeological evidence, and in the conventional narratives of human evolution. Tools are preserved, weapons are preserved, structures are preserved. The specific daily relationship work of an experienced older woman, the mediation of a conflict before it fractured a cooperative alliance, the memory of which family owed which family a food debt from the previous dry season, the knowledge of which younger woman was struggling with her new infant in ways that would benefit from attentive support. This work leaves no archaeological signature. It is preserved only in its consequences, in the survival of the children it sustained, in the coherence of the social groups it held together, and in the demographic patterns it produced across the forager populations that have been studied in sufficient depth to make the pattern visible. personal DNA connection, BOM03. The specific cognitive and social capacities that this work requires, and that the neuroimaging research described in the previous chapter shows the menopausal transition specifically building, are capacities you carry in your body right now. The enhanced theory of mind processing, the improved long-term social memory, the faster cortisol recovery following social stress that makes it possible to be fully present in emotionally demanding situations without being destabilized by them. The elevated oxytocin receptor sensitivity that makes social bonding and caregiving intrinsically rewarding rather than effortfully obligatory. These are not abstract neurological measurements. They are the specific biological substrate of the social coordination work that postmenopausal women have been performing in human communities for 300,000 years, and that your nervous system has been building toward across the decades of your reproductive life. The invisible economy is not invisible in your body. It is written in the specific way your nervous system processes social information, manages relational complexity, and finds deep satisfaction in the care and coordination work that the world consistently undervalues, and that biology has been specifically engineering for longer than recorded history has existed. The second mechanism through which post-reproductive killer whale females prevent mortality in their offspring, and its human parallel, is social mediation. Research on killer whale intragroup social dynamics found that post-reproductive females play a disproportionate role in reducing socially inflicted stress on younger group members, specifically in reducing the competitive behaviours that produce stress hormones and physical altercations within the pod. Their presence acts as a social buffer, moderating conflict and maintaining the cooperative relationships that the group's survival depends on. When they die, the social fabric becomes more competitive, more fractious, and the increased cortisol burden of living in a more competitive social environment contributes measurably to the increased mortality rates observed in surviving group members. In human forager communities, the social mediation function of postmenopausal women is documented in the ethnographic literature as one of their most consistent and most cross-culturally generalizable roles. They serve as inter-family mediators in disputes over resource allocation. They are the primary maintainers of the reciprocal food-sharing norms that bind forager communities across household boundaries. They manage the social memory of who owes what to whom across the network of mutual aid that makes small group survival possible. In every documented forager society with sufficient demographic depth to examine this question, the presence of active, healthy, postmenopausal women in the community is associated with reduced intergroup conflict, higher rates of cooperative food sharing, and more stable social alliances between family units. Their absence is associated with social fragmentation, increased conflict, and reduced cooperative investment in the public goods, birth attendance, infant care, ecological knowledge sharing, that no single family can maintain alone. The invisible economy of post-reproductive women is not a minor social supplement to the visible economy of hunting and foraging. It is the organizational infrastructure through which the visible economy functions at the level of effectiveness that made the human species viable in the specific ecological and demographic conditions of the Pleistocene. And it runs on biological machinery that the menopausal transition specifically builds and activates at age 51 on a schedule that 300,000 years of selection has determined is the precise moment when the machinery's activation produces the maximum evolutionary return. Sit in the specific quiet of a late Pleistocene evening somewhere in the East African Rift Valley, a place that would eventually become the site of Jebel Irhoud in Morocco, or the Hadza homeland in Tanzania, or any of the dozens of archaeological sites across the African continent that hold the skeletal evidence of the women who built the species you belong to. The light is failing. The group has gathered near a central hearth. The children, 6 of them, ranging from an infant in arms to a 10-year-old moving at the edge of the firelight with the specific restlessness of children in those years, are distributed among the adults with the casual ease of a group accustomed to distributing its young. Not every child is with its biological mother at this moment. The infant is with its mother. The others are variously engaged with aunts, older siblings, and one specific woman who sits close to the fire with the particular settled quality of someone who has been in this landscape long enough to know that this specific hour of this specific evening calls for exactly this: sitting, watching, being present in the way that only comes from having watched enough evenings to know what each variety of quiet means. She's in her late 50s, perhaps 60. Her postmenopausal biology is operating at full capacity, not declining toward it, operating from within it, in the years when the renovation described in the previous chapter has completed its most disruptive phase, and the rebuilt architecture is running in its full intended configuration. Her social cognition is operating with the enhanced theory of mind processing that neuroimaging research documents in women at her stage of the life course. Her stress regulation is operating with the cortisol recovery speed that makes her capable of managing the emotional intensity of the group's interpersonal dynamics without being destabilized by them. Her ecological knowledge, accumulated across 40 years of foraging in this specific landscape, in every seasonal and climactic variation it has produced in her lifetime, is the group's most comprehensive single navigational resource for the question of what to do when conditions shift. She's also doing something that the grandmother hypothesis data captures, but that no model of evolution fully conveys. She's being someone that the group organizes itself around without anyone having consciously decided to do so. The younger women in this group do not defer to her through cultural convention. They orient toward her through the accumulated experience of having learned, across years of direct observation, that her assessment of complex situations is reliable, more reliable than any single younger perspective, more contextually rich than any recently acquired knowledge, and more consistently aligned with outcomes that prove to be correct than any other single input the group has access to. Evidence block. 4 converging data points anchor what this woman's presence means demographically. First, demographic analyses of forager populations estimate the average post-menopausal lifespan in forager societies without modern medicine, without the nutritional surplus of agriculture, without any of the life-extending technologies of the past 10,000 years, at approximately 26 years. 26 years of post-menopausal life as the biological baseline, achieved in the hardest possible survival conditions. This is not a few final seasons of diminishing contribution. This is a second adulthood, biologically sustained, directed entirely toward the group's future. Second, studies of the Hadza find that postmenopausal women's foraging efficiency, calories acquired per hour of work, is higher than that of any other demographic group. Because the caloric value of their knowledge-guided search for high-yield underground food sources exceeds the caloric value produced by the less experienced foraging of younger women per comparable time investment. Third, the Agta Forager data, examining 78 children and 29 grandmothers across demographic records from the northern Philippines, found that grandmother proximity was a significant predictor of grandchild survival outcomes, and that the specific form of grandmother contribution that mattered most was not financial or material provisioning, but the time-intensive presence and knowledge transfer that only a grandmother with accumulated foraging and social experience could provide. Fourth, computer simulation studies modelling hunter-gatherer group survival under varying resource conditions consistently find that the presence of post-reproductive females with high ecological knowledge scores significantly improves group survival probability during resource shortfall periods. Specifically, the periods when the group's current knowledge base, representing only the years of experience that its youngest adults have accumulated, is insufficient to navigate conditions that fall outside the recent ecological record. Biological controversy. Bomb 03. The strongest challenge to the full adaptive menopause framework comes not from within the grandmother hypothesis literature, but from a direction that researchers are still working to integrate. The evidence that the menopausal transition is associated with measurable health costs in postmenopausal women, including increased cardiovascular risk, reduced bone density, and elevated dementia risk in later decades. Costs that seem inconsistent with a design that was supposed to produce peak social functioning. If menopause is optimally designed, why does the transition impose health costs that partially offset the social benefits? The response within the adaptive menopause literature is that the health costs of menopause in modern populations are substantially different from the health costs in the Pleistocene environments in which the design was selected. The cardiovascular risk increase associated with estrogen reduction is significant in a population living sedentary lifestyles with high carbohydrate diets. In a physically active forager woman covering 9 to 14 kilometers daily with a diet of lean protein and fibrous plants, the cardiovascular risk profile of low estrogen is substantially less severe, and the decades of postmenopausal function that the design produces are less compromised by the health costs that modern environments amplify. Saist. Community location prompt 02. The post-menopausal women who held forager groups together across 300,000 years of Pleistocene survival did not know they were doing it. There was no word for what they were. They simply existed in the full expression of a biology that the species had been building towards since before language existed to name it. If you know a woman over 50 who's doing something that cannot be named, holding something together, carrying knowledge that no one else has, being present in a way that the people around her depend on without fully recognizing the dependence, leave a note about her somewhere in the comments. The evidence says she is doing exactly what her biology was designed to do. The 26 years are not a bonus. They are not a gift of modern medicine that evolution never anticipated. They are the operational period of the specific biological design that menopause initiates, the window in which the postmenopausal female's fully reconstructed social brain, fully available biological investment, and fully accumulated ecological and social knowledge combine to produce the maximum evolutionary return on 300,000 years of genomic engineering. The woman by the Pleistocene fire was not being kept alive by the group's charitable tolerance of her non-reproductive status. She was being maintained by the same evolutionary pressure that maintained the ovarian reserve depletion schedule, the 290 genetic variants, the DNA repair gene associations, and the 40-year post-reproductive lifespan of the killer whale. She was being maintained because removing her produced mortality consequences that selection could not afford. The 26 years are the product of the same design that produced the transition that initiated them, and the transition initiated them at 51 because that was when the accumulated experience, the freed biological investment, and the reconstructed neural architecture were all simultaneously available and fully operational. The resolution of secondary mystery 02, why the menopausal transition feels so disruptive if it is building something rather than ending something, is now possible with full evidentiary support. Begin with the evidence for what the transition is building. The neuroimaging research from Weill Cornell's 2024 brain scans studies found that estrogen receptor density in estrogen-regulated brain networks increased during the transition, rather than simply declining with the declining hormone levels. The brain was reconfiguring its receptor architecture, increasing its sensitivity to a signal that was becoming less frequent, as a radio antenna increases its gain to receive a weakening transmission. This reconfiguration is the neurological signature of adaptive response, not passive deterioration. So it is the brain doing what brains under selection pressure do, adjusting the instrument to serve the function the organism requires, rather than passively registering the loss of a previously available resource. The grey matter evidence adds the next layer. Published research on brain structure across the menopausal transition found that specific regions show volume reduction during the transition, followed by partial recovery in the postmenopausal period, a trajectory that researchers describe as consistent with compensatory neuroplastic processes. The brain loses volume in some areas during the transition. It regains volume differently afterward. The rebuild is not a restoration of what existed before. It is a reconfiguration toward different connectivity patterns. A brain that processes certain types of social information differently, allocates certain types of attentional resources differently, and accesses certain types of long-term memory differently, in ways that are consistent with the enhanced social coordination capacity that the postmenopausal years require. The white matter hyperintensities that research finds at higher rates in postmenopausal women, bright spots in brain imaging indicating regions of altered white matter, were initially interpreted as straightforward evidence of brain deterioration associated with menopause. More recent research suggests a more complex picture. Some white matter hyperintensities represent the structural correlates of the altered connectivity patterns that the postmenopausal brain develops. They are not simply damage. They are, at least in part, the imaging signature of connectivity reorganization, occurring in the white matter tracks that link the brain regions involved in social cognition, long-term memory integration, and prefrontal limbic coordination. The prefrontal limbic interface is where the renovation's most functionally significant product is most clearly visible. This interface, the communication architecture between the prefrontal cortex executive control systems and the limbic system's emotional processing systems, shows measurable changes in postmenopausal women that are consistent with enhanced top-down emotional regulation, the capacity to experience full emotional depth while maintaining behavioral coherence and decision quality under conditions of high interpersonal stress. Premenopausal women show stronger limbic reactivity to social threat signals, and somewhat slower prefrontal regulation of that reactivity. Postmenopausal women show a pattern in which the prefrontal modulation of limbic responses has become more automatic, more rapid, and more effective, producing a specific functional profile of emotional wisdom rather than emotional detachment. The emotion is fully present. The destabilization by emotion is reduced. This is precisely the neurological profile required to be the calm, experienced center of a birth network, a social conflict resolution process, or a group decision-making event under resource stress. Scientific Debate Invitation 03. The interpretation of all of this neurological evidence remains actively contested within the neuroendocrinology and cognitive aging literature. The critical question is whether the postmenopausal brain's altered connectivity represents an adaptive reorganization toward enhanced social coordination function, as the evolutionary framework suggests, or whether it represents the best neuroplastic compensation available in a brain dealing with the loss of estrogen's neuroprotective effects, a brain doing as well as possible with reduced resources rather than achieving a genuinely superior configuration for specific tasks. The honest answer is that the available evidence does not yet fully distinguish between these interpretations, and both are consistent with significant portions of the neuroimaging data. What the data does not support is the simple deterioration narrative, the idea that the postmenopausal brain is simply less functional than the premenopausal brain in straightforward ways. The pattern of changes is too selective, too directionally consistent with social coordination capacity, and too clearly correlated with the enhanced social performance that forager demographic data documents in postmenopausal women to support the simple deterioration model. The resolution of secondary mystery 02 is this. The menopausal transition feels disruptive because it is disruptive. It is dismantling the reward architecture, the hormonal sensitivity configurations, and the attentional allocation systems that serve the reproductive years, the competitive edge toward mate selection, the intense focus on one's own reproductive success, the hormonal sensitivity calibrated to pregnancy and nursing, and replacing them with the social coordination architecture that the post-reproductive years require. The dismantling and rebuilding occur simultaneously. The construction dust is real. The hot flashes are the hypothalamus adjusting to a new thermoregulatory set point after decades of estrogen modulation. The sleep disruption is the circadian system recalibrating under new hormonal conditions. The cognitive variability is the hippocampus and prefrontal cortex reorganizing their connectivity during the transition between two different functional architectures. These are building costs. They are the price of a renovation that takes, in most women, between 2 and 7 years to complete, and whose product, when the renovation is finished, is a brain specifically equipped for the most important social coordination work the human species produces. Now the 70% resolution of the primary mystery. Menopause at 50 was never an accident because the genomic architecture controlling it is too precise to be accidental. The evolutionary selection pressure that maintained it across 300,000 years was too consistent to have been operating on a neutral trait, and the specific biological functions it enables, the grandmothering investment, the reproductive conflict resolution, the social mediation, the ecological knowledge transmission, the neurologically reorganized postmenopausal brain, are too convergently documented across independent species, independent forager populations, and independent scientific disciplines to permit the interpretation that they are byproducts of extended lifespan, rather than the designed purpose of the transition that created them. The complete answer to why 50, why that specific coordinate, is what the conclusion assembles from all 10 chapters of evidence. The full weight of everything built across this script is now available for the question to receive its complete answer. The question this script began with was not gentle. It did not ask whether menopause might have some evolutionary basis. It stated, without equivocation, that menopause at 50 was never an accident, that it is a 300,000-year design. And it promised that the evidence assembled across 10 chapters would make that statement specific, would name the mechanisms, document the data, describe the neurological architecture, and deliver a complete answer to the question of why this transition exists in the human female body at this specific age rather than later or earlier, or not at all. The complete answer is this. Menopause at 50 exists because approximately 300,000 years of human evolution and a parallel evolutionary process in 5 species of toothed whales, independently running the same selection experiment in the open ocean, determined that a post-reproductive female who redirects her full biological investment from individual reproduction toward group survival infrastructure is, across the long arc of generational transmission, more evolutionarily effective than a female who remains fertile until near death. This is not a warm sentiment dressed in scientific language. It is a specific mathematical outcome, tested and confirmed across thousands of generations of forager group survival data, encoded into approximately 290 genetic variants, controlling the transition's timing, heritable at approximately 44%, and maintained under active selection pressure for as long as Homo sapiens has existed as a species. The timing at 50 is not arbitrary. It is the specific age at which three conditions converge simultaneously in the female body and social context. First, accumulated foraging and social knowledge has reached its maximum operational value after approximately 30 years of direct practice in the specific landscape and social environment the group inhabits, making the post-menopausal, woman's contribution to food acquisition and social coordination more effective per unit of biological investment than any contribution she could make through additional personal fertility. Second, the reproductive risk of continued fertility has increased to the point where the mortality cost of additional late pregnancies begins to subtract significantly from the caloric and social contribution that the woman's survival enables, making the genomic calculation favour cessation over continuation. Third, the specific social dynamics of intergenerational coexistence, documented in the reproductive conflict hypothesis, have generated conditions under which removing the older woman from fertility competition creates cooperative space that is worth more to the group's survival infrastructure than the additional offspring she could produce in competition with her daughters-in-law. 50 is where all three calculations converge. Not 45 because the knowledge accumulation is not yet deep enough, the reproductive risk not yet high enough, and the social conflict costs not yet high enough to justify the transition at that age. Not 58 because the delayed transition would impose years of reproductive competition and resource conflict that the cooperative social system cannot absorb at acceptable cost. 50 is the coordinate. The genome knows it. The DNA repair genes that control the ovarian reserve depletion rate are calibrated to arrive at that coordinate across the full range of human developmental variation. The 290 genetic variants maintain the coordinate's location against the continuous evolutionary pressure that would drift it in either direction if the coordinate were not the optimal solution to the selection problem it was built to solve. Now the personal dimension, which the evidence requires to be stated plainly. You carry this design, every aspect of it, the 290 genetic variants, the specific DNA repair gene architecture, the ovarian reserve depletion schedule, the neurological renovation machinery, the postmenopausal brain's enhanced social cognition configuration is operating in your body right now, on the same schedule that operated in the body of every woman in your direct maternal line across 300,000 years of human existence. You did not inherit this from a species that was slightly more careful with its resources. You inherited it from a species that survived an 800,000-year-old population bottleneck in which 98.7% of the genetic diversity of the human lineage was lost, and in which the survival of the remaining 1,280 effective breeding individuals, and therefore the survival of every human being who has ever lived, depended on the specific biological capabilities of postmenopausal women doing exactly what the design was built to make them do. If you are approaching this transition, the evidence in this script is not reassurance. Reassurance is too small a category for what the data supports. The data supports something considerably larger. The recognition that the biological transition you are approaching is the scheduled activation of the most sophisticated social brain configuration the human species produces, a configuration that took 300,000 years to engineer and that your body has been building toward across the entire span of your reproductive life. The disruption of the construction phase is real. The heat and the sleeplessness and the cognitive reorganization are the authentic costs of a genuine renovation. But the renovation is not a metaphor. It is occurring in specific brain regions, in documented connectivity patterns, in measurable receptor density changes, as in the neuroplastic recovery of grey matter that is growing back differently from how it was before, growing back configured for the work that the postmenopausal years are designed to perform. If you are past this transition, the evidence is a specific form of recognition that the culture you live in has not yet learned to offer. what you experienced, the disruption, the reorganization, the profound sense of something ending that turned out to be something changing, was not your body failing a test it should have passed more gracefully. It was your body doing the most structurally demanding thing it has ever done, and what it built in the process, the social acuity, the emotional regulation, the long-term knowledge retrieval capacity, the deep satisfaction in the caregiving and coordination work that your nervous system is specifically configured to find intrinsically rewarding, is the functional expression of 300,000 years of selection working correctly. The killer whales in the Pacific Northwest who lead their pods to food during the hardest years do not know they are demonstrating the adaptive value of post-reproductive female expertise. The Hadza grandmothers who spend more hours per day digging tubers than any other person in their population do not consult the grandmother hypothesis literature before deciding how to allocate their energy. The post-menopausal women in human communities across every continent and every documented culture who hold the relational networks together, transmit the accumulated knowledge, attend the births, resolve the conflicts, and organize the cooperative infrastructure that no younger person has yet lived long enough to organize, none of them do it from evolutionary awareness. They do it because their biology is running a program that was written before their grandmother's grandmothers were born, and the program is doing what it was built to do. You are that program running. Not in diminished form, not in the shadow of what you were before, in the version of yourself that 300,000 years of the hardest selection pressure the species has ever survived was always building toward. The transition is not the end of the story. It is the point where the story becomes what it was always designed to be. Some things, when they are fully understood, do not need to be acted on immediately. They ask only to be held, in the particular stillness of a late night, in the space between the day that just ended and the sleep that is coming, and allowed to settle into the understanding they deserve. You are not a body in decline. You are not a design that ran out of purpose at 50. You are the specific biological answer to one of evolution's most demanding questions, running on a timeline that was set before you were born, building something that the species has depended on for longer than any civilization that has ever existed. The design is complete. It was always complete. It was waiting for you to reach the stage where you could feel its full weight. And now you have.